What's up, APBio Penguins? So tonight is Unit 7 Q&A with the APsolute RecAP. So we're waiting for Melanie to log in and get all logged in. Remember, if you're not following the APsolute RecAP, definitely you want to go ahead and follow the APsolute RecAP. She is a podcaster. And so what happens is she's actually has like little 10-minute podcasts over a variety of topics, but biology is one of those topics, as well as chemistry and government. And hi, Melanie.
Hi.
So she's going ahead and giving them the lowdown about your podcast and how amazing you are.
Thank you.
So hi, welcome everyone. I'm Tiffany Jones from APBio Penguins. I'm just a lowly teacher who just decided to throw some review stuff online. And you are?
I'm Melanie Kingett. I am the co-founder of the APsolute RecAP, and I am the host of the biology podcast.
Okay. So tonight we are doing Unit 7, which Unit 7, if you're not sure, like the order of the CED and the order in the different units, this is all about evolution and natural selection and all that huge chunk, which is a big, if you look at all the percents of the CED, this is actually one of the larger sections. It's Unit 3, which is like cell energetics, and then Unit 7, which is on evolution. And
so
like, I think they actually call it natural selection. But
natural selection, yeah. When do you teach this unit in your sequence?
It's the second to last unit. I actually go in order. I'm one of those teachers.
It's interesting because I also go in order, but in my
let me pause you for a second. While we're kind of reviewing a little here, if you have any questions, you can go ahead and put them down that little question box. It looks like a little question mark. We're almost just kind of getting ready and letting you have time to type your questions. So make sure you're putting your questions down there. Go, Melanie.
Okay, great. So in my non-AP course, I actually teach evolution like really early on, like second. A really great quote that always stuck with me, and I'm going to misquote who said it, but it's nothing in biology makes sense except in the light of evolution. So if you don't know evolution first, then discussing all of the other factors that are influenced by evolution or influence evolution, they kind of like don't have as much meat on their bones. However, I mean, you and I both, we start talking about evolution early on anyways. We can't help ourselves, right? We're talking about differences in amino acids and why they're different. We kind of like itch to discuss it.
Yeah, and as I go through these different things, I actually mention, so this will come up later when we talk about this, and I find it as like a scaffolding tool. Like they know their evolution's coming, and they have a general idea of what evolution is, just from previous years. But I don't officially do it till the end.
And typically and unfortunately, some misconceptions about evolution are pretty pervasive by the time we teach about it. So
okay, so it looks like we have already 5 questions in the box. You ready to rock?
Let's do it.
Okay, so I'm going to do my goal to always try to remember to ask the question before we start talking about it, because when the live is downloaded as a YouTube video and pops up online, students that are watching don't see the question. So we just start answering questions like, what's going on? So the question is can we go over analogous versus homologous structures?
That's a great question.
Now somebody just types in the chat. Remember, questions have to go in the question box or we won't answer them or we won't see them to answer them. So put that in the chat. The question box.
And so my approach to all these, and even the one that popped up in the chat about speciation, is just to use your root words and then think of an example that can carry it through for you, like when you start to see an unknown. So wings, right? Wings always come up when we talk about homologous and analogous structures because they can kind of fall into both categories depending upon the organism you're referencing. Right. So if we think about wing structure and look underneath the wing, right, at the bone structure, you could be talking about homologous structures because it has the same pattern, like 1 upper, 2 lower, and then a series of radiating bones. And so their functionality is similar for flying, but so is their structure underneath it all. And so that's going to suggest common ancestry. Now, why, Tiffany, don't analogous structures have as much weight under them for that common ancestry?
Analogous structures come from convergent evolution. And so they're individuals that have gone through the same selection. So they had a similar environmental factor that caused for the selection of that trait. And so similar problem, similar solution. And so just because they have the same solution doesn't mean they're actually related to each other. A great example is the flying squirrel and the sugar glider. One of them, I believe is the sugar glider, is a marsupial. And so if you know anything about marsupials, they actually are from Australia, and they have a premature birth, and then the embryo fetus develops within a pouch, versus the flying squirrel is placental, and it actually stays in utero for the entire development and then is full term when it's born.
Yeah, and so
big difference you see there.
When we say related, we mean recently related. Obviously, everything's related at some point. And so this concept of like getting off the ground was an advantage so many times in the course of evolutionary history and living things, and that's why you see flying repeated over and over and over again, or gliding, or jumping, or.
Okay, so. Next question we have is, what are the 3 types of selection? So I think what they're focusing there is that disruptive, stabilizing, and directional. Yeah. So I think those are the 3 they're asking about.
So think of your graphs, really, when you think of these three, and there's a really great POGL for the types of selection. I don't know if you've ever used it in your courses before. Yeah. And it's very Googleable, unfortunately, for us to, I don't really care. I don't grade them. I want the students to work through them. So it's really great POGLs for graphing, showing where did the trait shift from, we'll say, the norm. And the norm can be varied for different examples. And so, in disruptive, you're going to end up with like 2 separate humps. I guess is my best example. You're splitting from the center into 2 humps. Sure. Directional is going to shift to one
You're
side.
disrupting the norm is what you're doing.
The norm, right. I can see it. That's like an M. Directional.
You are so side. Visual.
I can't help it. And the irony, right? The irony of a podcast lacking visuals, right?
Of course.
And then stabilizing, right? Getting closer to the norm and losing those extremes from your phenotype.
So always when we see questions about this, they always are talking about like giving them the verbal that goes with it. They sometimes won't give them a graph. And so you need to be able to pull out the verbal part of that. So when we see like directional, right, it could talk about that there was one type of phenotype. So let's say, I don't know, the moths, the pepper moths.
Pepper moths.
And so due to this change in the environment, now the trees are darker. What would you expect to happen to the population of moths? You would expect the moths to become a darker shade because of the fact that now they blend in more easily. So you would want to give some type of verbal that goes with it. And the directional, the beaks from, Darwin's beaks.
Beaks.
So there was, of course, that drought that caused the median beak to decrease because of the fact they didn't have enough food. And you're welcome, Samantha.
Samantha. Hi, Samantha. And I think about it, green thumb.
Yes, all of y'all, thank you. And then the stabilizing, we stabilize at that norm because there's something that's actually favorable about it. So human birth is a great example of this. Like if the baby's too small, doesn't survive, the baby's too big, doesn't survive, it can't get out. All that fun stuff. So great. Thank you for helping there. I'm bad at this whole finding things.
And just to be clear, like directional, any type of those selections, it's not forever. It's for the moment when the environment has those selective pressures. So even if you have directional selection, and the pepper moths is a great example of that because of the Industrial Revolution, right? They went directional towards one color, and then you put scrubbers on all the smokestacks and all the trees get cleaned and they go directional the other way. So it can shift over time, and it will likely shift over time.
Right. And so you just mentioned selective pressure. So I decided I'd throw this question in there. It says, what are selective pressures? So selective pressures are literally just what is going to allow for something to be more favorable or less favorable, right? So if we go back to pepper moth, right?
Yeah.
So initially, there's always been dark moths and light moths in that population, right? Just because the genetic diversity, there was this difference, okay? Now, there was a higher chance and a higher frequency of that light allele because the trees were that lighter shade, right? So the selective pressure here is we need a lighter shaded tree because if you're, you need a lighter shaded moth because if you blend in, you're more likely to be selected.
Not eaten.
Right, not eaten. Selected for. And then when the trees changed color and became darker, now the lighters are selected against. That's a selective pressure. So it's what is happening in the environment that's causing one to be selected for or selected against.
And selective pressures can be biotic or abiotic. So it could be something about like a prey situation, a predator-prey interaction, but it can also be you know, availability of sunlight or soil nutrients or, I mean, you name it. So this kind of links to ecology, which we'll both do with kicking and screaming next week.
Not next week, 2 weeks.
2 weeks. Oh, 2 weeks to kick and scream. We're just not ecology people, but we will do it for you guys. And so because they can be biotic or abiotic, you know, they aren't really always like, oh, if this happens, that happens. It's not always going to be a concrete answer because it's all so environmentally specific.
And then also that selection could be sexual selection.
Oh, yes.
So although you were mentioning predator and prey, which is a great thing, when we think about the those guppies, right, they have all this bright color on them that's going to cause them to be eaten, right? So the brighter colors you have, the more likely you're going to be selected to mate with. But you're also more likely to be selected to be eaten. So there's also this selective pressure of who gets to mate because they're surviving and who gets to mate because, well, they didn't survive.
Do you want to talk about fitness since we were mentioning reproduction, or you want to pop up another question?
We can talk about fitness. So fitness, okay, let me just let you know, students, the words survival of the fittest, bad statement. And saying that an individual evolved, also bad statement. Neither of those statements should be given, as well as also be careful of Lamarckian. Hey, Miss Goffman, you got to be careful for Lamarckian statements. So those like three things, and I've actually forgotten what our question was. We
were going to talk about fitness. So fitness actually comes up first in Unit 3. It's like that oddball, like piece at the very end of Unit 3, but it's talking about like molecular influence on fitness phenotypically.
Oh yeah.
So hi.
It's such a delay.
Yeah, I know. So when we talk about reproductive fitness, what does that imply? You said Lamarckian statements, so we might have to dive deeper into like what that means. So when you say reproductive fitness, how does that relate to molecular fitness?
I'm not understanding where your brain was going in this question, so why don't you just go ahead and answer, because I'm not sure where we're going. So I was thinking of the fitness in terms of a like biological standpoint of that an individual that is more likely to survive is more likely to reproduce and leave more offspring. So are you talking about the molecular, like the genotype part?
Yeah. So in 3.7, I think it is, it mentions like molecular fitness in terms of like the context of the influence of the different genotypic combinations and how in that
3.
Yeah.
We're talking about genotypes in 3 when you just cellular energetics.
Let me check the exact phrase. And
you don't have to. It's fine. I believe you.
Go ahead.
Anyways,
you keep going.
I don't even know which one we go with, but whatever.
Let's talk about I'm gonna read the direct quote. Here we go.
Oh God.
Variation in the number and types of molecules within cells provide organisms a greater ability to survive and reproduce in different environments.
But that doesn't specifically talk about genotypes.
That's true. I was extrapolating.
You are extrapolating, which is fine. And we will see that the genotypes and the phenotypes will change, as a student asked me earlier today, like, how does the genotype and phenotype change? Well, if it's more favorable, then the genotype's going to go towards that genotype. I don't even know where we're going. Oh, Miss Kaufman's doing my job. I love it. She's answering the questions in the chat. I asked her why she didn't do that last time. She
wasn't here. All right, so the answer in the chat is that it's about photosynthesis and cellular respiration. So lots of people have taught about C4 and CAM plants there.
But C4 and CAM is not required. It's an illustrative example.
It's not. Yes.
Yeah. Okay. Sorry, we're going to go back to the
move forward to questions. Here we go.
Okay, so how about we do a deep dive over here, because let's talk about Lamarck and then jump into Hardy-Weinberg, right?
Our 5 fingers, I assume.
Our 5 fingers evolution. But let's first talk about Lamarck, because I don't want to forget to mention that, because it's such a bad thing to talk about. Okay, so Lamarck is use and disuse. Okay, so if you use something, then you'll be able to pass that trait on. If you don't use something, you don't pass it on. Easy peasy, right? Well, that's not true. It's not the use and disuse component of it. Okay, so the example that we use is a giraffe, right? So a giraffe stretched his neck, and so since it stretched his neck, it got a longer neck, and thus was able to get to the leaves. And so its offspring, who came from the stretched necked giraffes, also have long necks. No, it had to do with because there was a mutation that gave a longer neck to a giraffe, and that mutation allowed the giraffe to get more food. That giraffe got more food, was able to survive, and was able to pass on traits. Okay, so the first example where we say that it stretched its neck, that's the Lamarckian statement. You need to be very careful about not saying that since something used something or didn't use something, that's why we saw the change in the population, was because something used or didn't use. Okay, you have to talk about that the population of bacteria, if we're talking about bacteria, the population of bacteria had a resistant allele, and due to having the resistant allele was more likely to survive in the presence of this antibiotic. And because they were able to survive, they were able to leave more offspring. So we saw a change in the population of bacteria to become more resistant.
Right. It's never going to be about what one organism does or didn't do. Like that's just not going to work for the whole crowd. Whenever my students in the initial stages start to get frustrated with the giraffe example, because you can talk yourself into thinking it's accurate, right? You're like, but they're getting taller. It's like, well, like if two people like worked out at the gym every day and got like muscles on muscles on muscles, they're not going to have like super muscular babies. Like that just sounds absurd. They're going to come out like all jacked up. And when you talk about that, they're like, oh, okay, well, I guess that makes sense.
Or you lost a finger in an accident during your lifespan. Did your child have a missing finger?
Right.
No. And always remember also it's in the genotype of, or the genes of the gametes. So just because you had a mutation in your lifespan that caused the change doesn't mean it happened in your gametes. Okay.
Yeah.
So let's talk about Hardy-Weinberg. So what are the 5 fingers of evolution you mentioned a second ago?
Uno, dos, tres, cuatro, cinco. All right. So we have the pinky. The pinky stands for small population size.
You talk about this differently than I do. Continue.
Oh, well, I do. Okay, so I do.
No, you're fine. You're good.
What
you do yours.
Do?
No, do yours.
All right, then we got our ring finger for mating, so that'd be non-selective mating, and then we have our middle finger for mutations, and then we have our pointer finger for wandering, so emigration or immigration, and our thumb is natural selection. Okay, so then it depends upon how you're gonna reference it. So are you talking about things that like cause evolution or things that don't cause evolution?
That's so if we're doing differences,
right? So
I talk about the way the population needs to be for there not to be evolution, and you talk about the way that things that they have to be in order for evolution to occur. And that's why we're different, which is fine.
So do you teach Hardy-Weinberg equilibrium first, or do you teach it after?
I teach this first, and then I talk about the math.
Okay.
Yeah. So for me, I do the no-no-nos. So I need to have a large population size. I need to have random mating. I need to have no mutation, no gene flow, and no natural selection. I'm a no-no-no.
Can we go, Samantha? Can we go over both?
I just went over the second one, Samantha.
It means the same thing. It just it comes down to your like remembrance device. So and each one can be treated individually. When you're discussing, they don't all have 5 have to be occurring for evolution to occur. So are you drawing something? What's happening?
I know it's going to come down next to my brain, so I'm going ahead and getting ready for what I'm about to talk about, because I have to write it backwards for it to show up correctly.
Oh no. Okay, so Hardy-Weinberg Equilibrium.
We're good.
Okay, it's going to be working through our frequency of alleles and how they change over time. So there are 2 equations. They are both on the equation sheet. And they both equal 1. Tiffany is writing them down in a mirror image. Let's see how this goes.
No, it's P's
and Q's, because P's and Q's are like impossible. And so what we have, just thank you. That's Azario. That's not. We did not come up with the 5 fingers of evolution. That would be both in biology. Paul Anderson.
There's a TED Ed video that's great. So please watch it.
Yeah, it's super good. It's only 5 minutes. It'll explain it. Probably much better than she and I just did. However, even for us, it's really useful to use these mnemonic devices to remember, did I catch all of my bases? Okay. So Hardy-Weinberg equilibrium has 2 representatives, 2 variables, P and Q, to represent the dominant and the recessive allele. Their frequencies must add to equal 1 within a population. And there we go. Keep going.
No, you keep going. I need to keep writing something.
Homozygous dominant typically is, and I say typically, is represented by P squared, homozygous recessive Q squared, and heterozygous 2PQ. Those values come from your Punnett squares. When we are expressing them and now solving problems, always start with your Q squared if you can, based on the information you have within the problem. You don't want to give yourself a circumstance in which you can't solve for something using a quadratic equation. So by starting with Q squared, you can solve for Q by taking the square root and then go back to get to, this is interesting. And it's like puppets when someone's trying to be your hands and you're explaining it. And then go back and solve for P by subtracting for 1, and then you can go and solve for P squared and 2PQ respectively. But don't just like do all the math without checking what the problem was actually asking for.
So I need to
go for it.
No, keep going.
No, because you may go 12 steps too far based on what the problem is even asking you for.
Okay. So that was all based on that they've given you one thing. So they were talking about the dominant phenotype, or they talked about the recessive phenotype, or they gave you the dominant allele, or they gave you the recessive allele. They've given you one thing. Okay. If they give you all of the phenotypes, they say there's 100 that are homozygous dominant, there's 200 that are heterozygous and 100 that are homozygous recessive, you need to use this formula to get your P's and your Q's, okay? So we're going to do 2 times however many I have that are homozygous dominant, plus the number that are heterozygous divided by the total number of alleles, okay? What that does is that allows you to find all of the A's, because I multiply this by 2 because there's 2 capital A's. I multiply this by 1 because there's only 1 capital A. And the same thing would go down here where I look at my Q, which was my recessive allele. 2 times my homozygous recessive plus my heterozygous will tell me where all the A's are. Okay, and that's what I want to make sure that we mention, because the math that you're going to see on the AP exam can be either one. We've gone through multiple examples this whole year about this. If you want some how do you do this math, I actually did a video like back in October. So if you flip through my IGTVs, you'll see one on Hardy-Weinberg. It is early in the game, which means that it is definitely, have you ever noticed that there's like cans of like veggies
because you're stacking
me?
Your camera.
Right. So back in the day before I figured anything out, I actually would stack my camera up, my phone up, so that I could do a live stream. So there's the cans still up here. I probably should check them downstairs because they're probably going to go out of date soon.
No, leave them. They're part of the fixture. Maybe they're expired.
Okay, so I hope you've answered this person's question.
We got a couple questions in the feed, guys. Just a reminder, if you can put your questions into the question box, we tend to lose them in the feed, but we will try to get to as many as possible. And reminder, we are doing Unit 7, which is a natural selection. And yes, the equation sheet, thank you for identifying that, is on in the front of the test booklet if you are paper. And if
do
you
not rip it out.
No,
you get big trouble.
Big trouble. And in the digital version, there's going to be a button for you to access.
Right. That was
you can flip back to it.
That's a big issue with students, and normally, because I guess like in teachers' classrooms, they let you tear it out, or they let you, have it separately. You cannot take anything out of your exam book if you're taking a paper exam. Okay. Yeah. So do not tear it out. There's a reason why it's not perforated.
Even if your proctor says okay, because your proctor is not going to be your science teacher. They can't be anywhere near you on the day of the test. And
sometimes proctors don't know the directions.
They don't.
So do not tear anything out. No. Okay.
All right.
So next question we have is, can you go over allopatric and sympatric speciation, which I think it was on the chat at the beginning, and like you mentioned the words, but we haven't actually gone over what they are. So let's go ahead and jump into those.
So allo.
Hi, Dan.
Hi.
Allo. He took it last year. I don't know why he's here.
Oh, did he? No
yes.
way. Dan hangs out on my chat all the time. I don't think so. Dan said he was. Oh, Dan's taking AP chemistry this year, if I remember correctly. Okay.
He's a junior in New York.
Allo, I hope that heart was from him. Allo means other, and we've seen that prefix before when we talked about enzymes. And so it's a really good kind of analogous thing here to use your root words to help you in both places. So if it means other, it's implying. So live 1 second. Oh, Dan, see if you can answer any questions in the chat. We'll test your knowledge. So allo means other. So we're talking about speciation that has occurred because they are in other places, like separated places, usually geographically. Sympatric is going to be the same space.
And so oftentimes with our sympatric, we're going to see that there's these reproductive isolation techniques, right? We have to find some way to inhibit two organisms from mating. And the big thing of what this whole comes down to is what is a species, right? So the biological species concept says that two organisms must be related if they're able to reproduce and produce fertile, viable offspring. It all comes down to, can they reproduce? If they can, are the offspring fertile where they can also reproduce, and also viable as in they're healthy and good and there's not going to be medical major problems. Like we're talking about overarching problems of the species, not like an individual itself. And so I lost where I was. Okay, so there's these reproductive mechanisms to kind of inhibit two organisms from mating. So these are honestly, like, you hear the name and you know what they are for the most part, right? Behavior. Well, they have two different behaviors, right? So it's a mating song, it's a mating dance, it's some type of ritualistic thing that the organism does in order to attract a mate. My favorite, it's a blue-footed booby.
Oh, it's so cute.
Those feet, they show off their little feet and they're like, hey, come mate with me because I have the pretty feet. I remind students that peacocks are actually the male, and that's why they're so beautifully colored.
I learned something
to attract
so cool about peacocks the other day that I never knew.
What, they're males?
No, not that they're males. That their coloration is not a pigment.
It's not a pigment.
It's not a pigment. It's due to light refraction. And so that's why it never fades. It's fascinating. All of the coloration for peacock feathers is not it's not melanin. It's not a pigment. It's not a protein.
That is really cool. Okay.
Isn't that cool? I'm sorry.
Sorry.
By peacock feathers.
Temporal
which is
isolation.
what they're supposed to do. They're supposed to distract you.
Temporal isolation is going to be different time. And so it's either different times of the day, different times of the month, or different times of the year, or like seasons. And sometimes they're alternating years. And so if they don't mate at the same time, of course, they won't mate with each other. There would be mechanical isolation, in which square peg, round hole, they just can't mate together. I'm missing some. Habitat isolation or geographic isolation, they kind of fall into the same boat there, where they don't live in the same area. So, of course, they don't mate with each other to mate. I'm missing one. What am I missing?
I lost track. I was in the chat. Sorry.
That's okay.
Very unhelpful.
It's okay. No, it's fine. We also have gametic isolation, which is where the actual gametes won't fuse. So although they attempt to mate with each other and the sperm and the egg are in the same world, this usually happens with external fertilization. The sperm will not fuse with the egg. Oh, now there's Kaufman coming in with the late game of the gametic. Thank you. Which you probably typed it before I said it, but it's the lag. And then that's all pre-zygotic, so before we make the zygote. So let's say sperm and egg do fuse together, we make the zygote. There's also reduced hybrid reduced hybrid viability, which means that it's not as fertile, it's not as viable, it's not healthy. Reduced hybrid fertility, which is now is going to be sterile, which a lot of my students didn't know this. Did y'all know that mules are sterile? Like when a horse and a donkey mate together, they make a mule. And
the only way you're getting another
sterile.
mule is to mate another horse and a donkey.
Right. They will never be. And that's why their fitness is actually zero, because they're unable to donate to the gene pool. And then we have the hybrid breakdown, which is where the first hybrid is perfectly fine, perfectly safe, no issues. But over generations of reproduction, it actually starts to break down and it is weak and feeble. So I think I've said all the post-zygotic ones.
Yeah. There's a really good example of the was it the mosquitoes in London, that when the underground was formed, they speciated because of the fact that they were separated by the tunnels. Yeah. So the human aspect of we can cause that speciation to occur more quickly with some geographic barriers.
I really want to get Kaufman to like turn her camera on. I want to see what three people look like on this.
Also, you can, I'm just saying, Kaufman, we could add a third to the live, maybe for exam review.
Yes, let's
ponder it. You can do like 3s and 4s now.
Yes, I just want to know what 3 even looks like. Okay, so that's all.
Referencing resource partitioning or referencing, no, it says no. So
I don't
again,
even
guys,
know what to do with this, but I'm just going to throw that one up there. Other mutation, what causes or drives evolution? Yes, she's on board.
what was she saying no to then?
Oh, I don't know.
It was a big no, and then it was sounds fun. All right, well, we'll just put a request in next time.
Okay. Other mutations, what causes or drives evolution?
Lots of things. I mean, mutations are only going to be what gives you your new genotypes within a population, but then after that, how those genotypes interact.
I got
Is
you.
that what I'm
Ready?
getting at?
You ready? What changes what causes evolution? Are you ready?
Yeah, let's hear
I have
it.
a really small population size, and a dramatic thing happens in my environment, and my population is reduced even more. That is going to cause evolution. If I have sexual selection and I select for a certain mate, like if I only want to mate with red-haired people, then of course, then I'm going to see a change in the red-haired population. That causes evolution. If I have a mutation, evolution. If I have gene flow, or where there's individuals flowing from one population to another, there's immigration. That causes evolution. Natural selection causes evolution. I really don't know where they're at. Yes, bottleneck happens with when we have a small
Small
population.
population size.
And actually, bottleneck is very dangerous because it can cause for harmful alleles to become fixed. So when we dramatically reduce our population size, certain alleles can increase in frequency, and then we won't have the more favorable alleles. So this kind of makes me think of like heterozygote advantage in a way. So y'all know that. Have you ever watched the HHMI video on sickle cell, y'all? Like, it is amazing.
So good.
It talks about, his Tony Allison, or I cannot think of the scientist's name, but it's okay.
I'm trying to think too. I can see him.
Totally. Back in the day, he was investigating sickle cell anemia, okay? And he was trying to figure out, actually, was he looking at malaria? I don't know what he was looking at.
He was looking at malaria, and then he realized what he was plotting, like, as it was going, and then he started taking more samples.
Right. So he saw a correlation between where there's a high chance of malaria and where there's a higher probability of sickle cell. And it comes down to that if you have sickle cell trait, like you are a carrier of it, then you're less likely to be affected by malaria, and you're also less likely to be affected by the sickle cell anemia characteristics. So it's something called heterozygote advantage, where it's more advantageous to be a heterozygote. You're still carrying that negative allele, but when two heterozygotes mate, there's a 50% chance of getting a heterozygote, which ends up making you a more favorable characteristic. If you're homozygous dominant, you get malaria, no problem, not good. If you have, sickle cell disease, not good.
Not good.
Now, again, this is all based on way back in the day. Like, this is not talking about like current days, right? This is talking about way back when healthcare wasn't what it is, or what it is now. So that's really what it comes down to. Okay, I'm dropping the question.
No, but the point of this is, you may get a question about malaria and sickle cell, and you say in your head, I've never learned anything about malaria. How can they, am I supposed to like cure malaria on this exam question? Absolutely not.
They had to kill cancer last year.
They did. I had to cure cancer last year. And that's kind of the whole piece of it. Like, if you expose yourself to as many, application examples as possible, kind of like put yourself out of your biological wheelhouse. And I know it's hard to do as a student, but your teacher has resources and can point you in the direction of more resources. And watch Tiffany's FRQ Fridays, because going through all the old FRQs where they're all application is a great strategy as well.
So my dog was very upset because Marco on Marco Learning always gets to have some screen time. So Bailey wanted to have her screen time.
Hi, Bailey.
She says she's a penguin, and she doesn't really understand why she can't be a penguin, but she's going to pretend like she's a penguin tonight.
So
okay,
good.
There's her screen time.
Hi,
we're going to let her go bye-bye. Say hi, Bailey. Bye, Bailey.
Winnie would not fit on my lap. It would be Winnie, and then her head would be up here.
Okay, so somebody asked about divergent and convergent evolution, and it's been since we mentioned that earlier, so I want to make sure that we kind of really make sure that we describe that. So divergent makes you think of coming apart, right? So this is our speciation, right? We had the one organism that went through some type of characteristic, some type of situation that caused it to speciate into two different. So this can either be rapid or it can be a gradual evolution. What's the rapid? Punctuated equilibrium.
Punctuated equilibrium.
Or we can have gradual equilibrium. Punctuated just means it's immediate change, and then gradual is kind of slowly.
And as might imply, that's influenced by environmental pressures. So was it a dramatic environmental circumstance, or has it just been like slow and steady wins the race here?
We have someone trying to jump in on our video.
Oh yeah.
We're getting bombed.
That's all right.
Okay, so then conversion is going to be dealing with that they're not actually related to each other. So as I said before, similar problem, similar solution. So they're not actually related, but they cause they look the same. So if you've done the Rock Pocket Mouse Lab, although these mice are all actually related to each other, and they've actually found that genetically they get the mutation independently of each other. So although they technically all are brown mice or black mice, they got a mutation that's a different mutation. And they're not actually coming from a single brown mouse. It's just they all had the same, they're dark. The only ones that can survive are dark. So there we have it.
And that kind of leads us to like just general evidence of evolution, how you go about designing your cladograms and why.
Talk about those. There's no question in the chat about those. So.
And so like why you end up with molecular evidence being stronger typically than morphological evidence or things you might see kind of externally of an organism, because from the outside, of course, they're related. But internally, when you look at their amino acid sequences, their mutations, their DNA components, that's really going to give you more information about their relatedness.
And Ms. Coffin mentioned down in the chat, she talked about the stickleback spine. Stickleback was actually on the test for like 4 or 5 years in a row. So it might make a comeback. It hasn't been there in a couple of years, but it was there for like 3 or 4 like years in a row. And I was like, it's going to come. It's going to come. And that never comes. So who really knows? So somebody says, this is actually, we keep getting off task in the chat. Like people are writing other questions in there. It says, why is genetic diversity important?
Okay. Well, you've talked about small populations. Did we talk about diverse populations?
No, we haven't talked about diverse populations.
I mean, diverse populations, if you're talking about the same species, which you should be, if you're talking about a population, is really going to make you more resistant to change. So if you have a bacteria population that's all the same and an antibiotic comes in and wipes everybody out, well, then that's not really successful for that organism group.
Which brings us into the type of reproduction too.
Yeah. And so what we end up happening is, in order for the perpetuation of a species to occur, more diversity is going to be advantageous. That means more organisms will be likely to survive when there are environmental changes. And it is the population that evolves over time. While we're talking about genetic diversity, just like biodiversity in general, so referring to not just one population, but multiple populations in a community, is also really advantageous for the health of that community. So this is a parallel explanation that you're going to see as you get more and more zoomed out within an ecosystem.
Okay. And someone asked in the chat about artificial selection. So like artificial selection is actually from humans. So like we breeded a certain dog type because it was a good hunting dog.
Bailey would not exist in nature.
No, we had to have that. Yes, so artificial selection is just we select for certain characteristics that we find favorable. Was it the mustard that gave rise to the kale and the cauliflower and the broccoli? All types of things because of that artificial selection. So humans have selected for a certain component, and that's why we call it artificial. It wouldn't actually have happened in nature. Same thing with corn. Corn used to be maize, and maize we select for a certain component of the maize to make the corn as it is now.
So Kaufman put a really good example in the chat about the potato famine and the blight infection that happened, which is why we had the Irish potato famine. But also, I'm very concerned for our bananas currently.
That's what I was going to tell you.
No, all of our bananas are identical, and it's the casa. No,
I don't remember the mute.
Begins with a C. Anyways, guys, our bananas are in trouble. If you are a banana lover,
and there is a type of fungus that is taking over that banana brand and like the banana species, and they cannot figure out how to get rid of it.
We will not have bananas as we know them in maybe 20 years.
And bananas are triploid. Okay, so those of you who don't know really can figure out why that all this happens, right? They're triploid, which means they cannot go through sexual reproduction. Okay, they're going through asexual reproduction, so they are all identical to each other.
Cavendish, yes.
Thank you. I had to do
Cavendish.
research on it when I was writing a question like 3 or 4 years ago on these bananas, and I was like, oh, I'm gonna write on bananas, triploid, and then I like the more I researched, the more I found that stuff out. I'm like, oops.
Yep.
So yeah.
So the Cavendish
Green
banana,
made a TikTok about it.
Of course he did. Yeah. So like
Thanks,
when we
Green.
think about our artificial selection in our food source, it's a huge association. Artificial selection in and of itself is not bad. I mean, we create food products that are resistant to disease, usually. They yield high crops. You know, it makes our food more efficient, but there is the downside. If you don't have genetic diversity within your population, you risk these types of things.
Okay, so students oftentimes have issues with the term sometimes. So thinking about the difference between genetic drift and gene flow. Because like when you think of drift makes you think of flow.
Movement,
so how do you keep them separate when you're explaining it to your students?
So when we talk about flow, we're always talking about like physical movement. So immigration and emigration. And so I mean, I haven't had to dive into the differences much with my kids. I teach far fewer students than you. What did your students need for this topic?
The way that I always try to explain it to them is like, I always come back with the founder's effect. And so when I explain about founder's effect, talk about these random mice just climb up onto a log and then they drift down the river, right? And then they find a whole new population on an island, which gives you the founder's effect, right? So there's a small population that gets isolated away from the original population. So that is how I kind of get the genetic drift.
You use the word drift literally.
I use the word drift literally just to try to help them out. Versus gene flow, I'm talking about that the genes are flowing into a population. So there's actually a population that's there and I'm gaining them or losing them in that flow. I don't really have much. So can I explain an application of it? So I think we just did an application. Yes, the live is going to be saved. It'll be posted to YouTube pending it doesn't crash like my FRQ Friday did on Friday, which I discovered, I think, why. I got an error in the middle of it that said that I was out of data. And I was like, what does that mean? And I ignored it. And then, well, that was bad. So I deleted like 20 million things on my phone so that hopefully this will not have problems. So, you know, stuff that happens when you try to do things live. Okay, so let's take a pause for a moment and let's, like, you posted today in the Facebook group about tips and tricks for the AP exam. What was some things that some of the teachers have said to you about, like, last thing to tell to their students?
Yeah, so I posted in the group and I said, what's your favorite piece of advice to give to students before AP exams? What I really loved about what came through is half was test taking tips and half was taking care of you as a person and as a student. And I think both are equally necessary. I think my favorite for actual test taking tips are don't leave anything blank, which you said, because you do yourself a disservice by not guessing, right? How you go about your guessing, I think that can be really diverse over time. And then FRQs, you don't have to answer them in sequence, in order. Like, you know, do the ones you know, go back to the ones you're less confident in. And these pieces of advice are specific to the paper test. Right. So obviously that may not be a strategy you can implement on the actual digital version in terms of bouncing around.
You could still implement that one though. You can answer D. If you really know D, go ahead and answer D, go ahead and answer B, and then come back to A, C, and E if you need to answer them in the order you know them.
Right.
So that
label them and label them. Yeah.
Yes.
And label them. So those were two I thought that were just like really practical. And then there was the other side of it. I saw this one I really, really liked. And it was, if you really don't know at all, if you have no idea what the answer to the question is, pick something related to evolution or cellular energetics, since those are most heavily weighted on the exam. And I hope that you don't get to that place. But if you're really stuck in guessing, that would be a strategy. But sleep, take a deep breath. It is one day. It doesn't define what you know about your biology knowledge at all.
And those of you who are still following me when the exam scores come out, I'm going to post something that literally will say, you're more than a number, because you are. This has been an entire year that you've learned something. And it's one day, one test. The test takers don't know what happened to you that morning. Maybe, you know, something traumatic happened on your way to the exam. Last year, I had a girl fall down the stairs on the way to her AP exam. Like literally fell down.
Concussed? Oh my goodness.
Concrete stairs, and was bleeding from arm to arm when she went in to go take her AP exam, having a panic attack because she forgot the exam was happening. So, you know what? You know, it's just one day. No one knows what's happening to you that morning. So you still have an entire year's of knowledge. Okay.
Yeah.
So somebody asked us, and we'll answer this, and we'll go back to some biology stuff. They said, would she recommend the in-person or the virtual if you're given a choice? I know that some counties are giving the students a choice and some counties are not giving a choice. So which one do you recommend?
I would recommend in-person and paper. Even if you've been learning virtually all year, I think that there is a comfort to being able to interact with it in a way that you have more control over. And not that you don't in the digital version, but you have more experience as a student taking paper tests and taking digital tests. And so there is a comfort in that experience, like broad spectrum. And I know it's so tempting. You're like, but the digital exams are later. I will have more time to study. I can promise you there's nothing you're going to learn between May 14th and June, 6th is the last test that will impact your June 11th, that will impact your score in a concrete way. Even if you haven't learned the last unit yet. And I say that, you know, with a crash course experience of unit 8, if you need to do that through some YouTube videos, your application knowledge that you know will get you through like most of unit 8, to be honest. And so I would choose paper if I could.
Okay.
What about you?
Oh, I hands down paper. But my reasons is what you said, yes, but also the test security, right? We know the test security, the test developers know the test security of a paper exam. They know you don't have access to any resources. They can tell you all day long, no accessing resources, but honestly, you and I both know there's no way to really stop a student from using a phone, especially since there's no cameras required, right? And even with a camera, you and I both know there's probably a student somewhere that's figured a way to use their phone. Okay. So I think, and I don't know for any, I have no knowledge of this. Okay. I believe that the digital exam is going to be written in a way that you won't be able to search things, or if you do search things, it's going to take you a long time to search them. And that's why they have the no go back rule, is because then you can't have students collaborating on the exam. And one student doing like questions 1 through 20, another student doing like 21 through 40, and then they work together and come up with it. If you're going to cheat, you gotta, I'm not gonna tell you how to cheat. Because I was always a devil's advocate. I can come up with how you could cheat, but let's not do that. But so they have to write it in a way that is going to inhibit you from cheating. And so I think that paper exam is definitely the way to go. I know it's less time to study, but are you really, really going to do anything between those days that's going to make a big difference? I've had students start the school year and make a passing score on the AP exam with it just being multiple choice. So they don't get the first points portion. They only get a multiple choice, and they've already been able to pass it. Okay. You don't have to be perfect on the exam. Okay. I think based on the practice exams, I think it's like a 50% raw score on the exam and you can pass. Okay. So don't overwhelm yourself, number one, trying to figure out all the answers. And number two, you got this. But I think that paper is definitely the way to go. Sorry, I kind of went off.
No, and I agree. And okay, so let's just assume you didn't have a choice. You are taking digital. If that's you, change your study habits yesterday. You want to give yourself that experience of only moving forward when you take a test. Don't go back and check your questions. You know, you want to make sure that you are timing yourself and pacing yourself the best that you can, because you're not going to really be able to like modify some of those things. The question was, are notes allowed in the online exam? So back to what Tiffany said, they're not going to be able to stop you from doing some things, but you are going to do yourself.
The official word is no.
You're going to do yourself a disservice by putting anything distracting in your area, because it's not going to help you, and you're going to waste time looking at things that aren't going to get you to the answer to the question, because they're going to be application questions. And so you're going to need to really dive in to what the question is asking you, and specifically tie that biological knowledge in.
Okay, so let's jump back into biology for our last 10 minutes. And it says, how does reproductive isolation lead to speciation?
Okay, so my argument would be, how does normal reproduction lead to lack of speciation, right? So if you think of it from the other perspective, if organisms can reproduce with each other, they're going to perpetuate the same gametic combinations.
Paul, will you put that question into the question box? I want to talk about that, but I can't talk about it right now.
And then if you refer to isolation, right, if you aren't allowing two groups, allowing for varying reasons Tiffany went over earlier, but if two groups are not able to combine their gametes, then they're going to lead to further and further differences between their genotypes later on down the line. Probably harder.
It was about don't worry about. Yeah,
okay.
I'll answer this question in a second. Were you talking about gene flow? I got distracted.
I was.
Okay, that's good. That's all I wanted to add in there.
Yep.
Okay, so I got that. A lot of the questions that are in our box are about other things. So this is our last. Yeah, there are other units. This is our last one that's about this unit. Remember, we're doing unit 7, which is on evolution and natural selection. So how can scientists use genomes to prove that all species continue to evolve? So the Hardy-Weinberg, that whole equilibrium is where we prove that evolution is taking place, okay? If your allele frequency changes, if your genotypic frequency changes, that means that evolution has taken place, okay? And then you could sit down and figure out which of the 5 conditions are not being met. And it's usually more than one that's not being met, but it's usually going to come down to natural selection because our environment's constantly changing, which then leads to the similarity. And then also the way that they can prove that individuals did evolve is they can also look at amino acid structures, right? That's one of the things we do with our cladogram, which we haven't mentioned, the phylogenetic trees and all that. So I'm just going to do a little plot in on that because I know that you're going to have something, right? Question 5 is going to be some type of drawing. And so the questions that we've seen before, there's been pedigrees. We've had phylogenetic trees, we've had draw on a cell. So there's going to be some type of drawing for question number 5. So phylogenetic tree is perfectly one of those. And so if you see 2 organisms that have a very small number of differences between them, they're going to be branched or close together on the cladogram or phylogenetic tree. The point where they come together, that's called a branch point or a node. They can rotate amongst that point. So don't get too freaked out. If you see there are 2 branches, it doesn't matter which one goes on which, just put them there, you're fine. The live I did, not this past Friday, but the Friday before that, I showed like 4 different ways you could have done the exact same
answer, right?
Yeah, and we had to, as readers, look for all of those answers because they were all plausible. So thanks for the person who wrote that question. And then if they're far apart from each other, then they would be, of course, far apart on the cladogram, and they would have a large number of differences. And that's what usually determines our outgroup, or the one that first kind of diverges off.
Basal taxa, yeah. And so that's going to be when you have more data instead of just like positive or negative character traits, which is going to lead you more on the cladogram side than a phylogenetic tree. Would you disagree?
You can do either one. They show the exact same thing. The only thing that I see that's different is in phylogenetic trees, we can do time. So there was the bear question 2 or 3 years ago in which they were looking at like brown bears and I can't remember the other bears that were on, and they asked you like how long ago was the common ancestor of these 2, and you had to actually look at that timeline, which caused a lot of drama for the readers because the axis was a weird, it was like tens of hundreds, and so students wrote it all kinds of wrong ways, so they accepted a lot of answers. As long as you were close enough, they let you have it. They were real nice on that question. Okay, so I guess let's just go back to this question because of the fact that the student asked about it and we don't have any more biology questions in here. So, well, unit biology. So they were asking about the digital exam and the paper exam and how I believe that they will be written to be ungoogleable and quote unquote, they're not really harder questions. They're just less able to be searched. And so just to clear everyone's minds, ETS, Educational Testing Service, they have some very smart people, okay? And these smart people analyze the exams, okay? And they don't just analyze one exam. They analyze every single question, Okay. And they determine the rigor of every question. And they determine if you know something on this exam. So let's say the first administration, if you know X amount of knowledge and you take the first one, you take the second one, or you take the third one, no matter what questions you get, you will get the exact same score because of the fact that they have these things called cut scores, and that is how they kind of scale the exam to determine what your score is going to be. And so they also can use your multiple choice score to predict your free response score. So when the readers are grading your free responses, if your score doesn't fall within a certain range, it causes a red flag, and we actually have to reread your free response just to make sure that we have scored it appropriately. Okay, because of the fact that we want to make sure that you get an accurate score. And last year, I think they even let teachers like knock it back and say, okay, well, they should have passed the exam. So I don't think that's going to be another thing that's happening this year. I don't really know. They haven't made that decision, but there is a lot of statisticians. They have a lot of patents. I've been to the ETS office and I've seen all their patents. They have a ton of patents showing off all of the analysis they can do on your exam. So I can guarantee you that no matter if you think your test is hard or if your test is easy,
it will be fair.
It will be equitable. And the questions also are written to be equitable. So they try not to make stuff like certain environmental, like, so if you live in a certain part of the world, you would expect certain, like, you would understand, I guess if you lived in Louisiana, you would understand that kind of climate, right? So they would give you background information in the questions to explain anything, right? That's why when we saw that mussel question way back when on the mussels about in the marine sound, they gave you all of the stuff you need to know to be able to answer that. They make all questions equitable. They go through, like, oh my God, like so many rounds of people between the person to write it and the people who edit it and the people who investigate it. They do so much investigation on your test to make sure that you will be fair and equitable. That is their top priority, is to make things fair. And so I know that sometimes y'all don't think that things are fair. I promise you, in the big scheme of things, we make it as fair as possible. So
yeah,
sorry,
so will it be harder? I think that, so content wise, no, it'll be fair. But the structure of the exam may be challenging for you, which may make it seem harder. And I think that's really going to be a personal experience. But let's just say you don't have choice. You have to take the digital exam. You got it. You know, start preparing out in the style that will make you best prepared for the exam. And that will give you a really strong footing for that experience.
And then, since we have no more biology, pacing on the FRQ. So if you are taking paper exam, spend 20 minutes on your long and I think it's 6 or 8 minutes on your short. That gives you more time to go back and kind of re-look through things and say, okay, I can add this in here, I can add that in there. If you are taking the digital exam, it is 25 minutes for your long and 10 minutes for your short. If you happen to be done with your long and you think there's nothing else you can add into it, it's perfectly fine to press the next button and go on to give yourself more time in some of your other questions. But don't leave anything blank. If there is still time on your timer and you are still on question 1 and you can come up with something. Do not leave anything
predict
blank.
questions.
Predict questions.
Especially predict questions. Just make a prediction. Increase, decrease, stay the same.
You have 3 choices there. Okay.
And
today's FRQ Friday that I did, there was 350, 350, 370, or 390. They gave you choices. Pick one. Don't care which one. Never leave anything blank. But so they said, you know, if we're running out of time, what do we do? Yeah, you can always leave things blank if you don't know. But skim through it, see if there's a draw, see if there is a predict. Because oftentimes on those predict or draw questions, you can at least come up with something, right? Try to find what you can answer. It's a points game, right? Where can you get the most amount of points? Just go ahead and throw it out. That's why I said that you can answer D first. If you know D, just go ahead and tell them D. We as readers, and yes, I am an AP reader, we as readers know that we have a rubric so memorized that you can give it to me in any order and I can still score it. I score thousands of them every year, so we're good.
There you go.
So any last minute questions about biology?
I can't believe, so we're already on Unit 7. We are going to come back with Unit 8 and then our exam review. In the meantime, obviously, you guys are going to start diving into a lot of things on your own. Please reach out to us if you have questions. We'll do the best we can to answer them either in DMs or in posts on our social media pages. We are here as a resource.
Right. So just make sure like we can say different resources depending on what you need, right? So is there a favorite test prep book you have?
I like Barron's, but my kids have all different ones.
Okay. So I prefer Barron's as well. I think Barron's is probably your best bet. Is there a podcast that you prefer?
I'm partial. I am partial. Now, speaking of Barron's, Barron's did just start a podcast.
But not biology.
But not biology. But if you're looking for other podcasts, yeah, Barron's did start a podcast.
Yes. TikTokers. I like Miss Sloan. Miss Sloan is great. So Miss Sloan Biology, she's got a great TikTok account. If you don't follow her.
What's the guy? Other TikTok?
A biology teacher.
Biology teacher. Yeah.
Yeah, he's sweet. He's nice.
Yeah, not as much AP content. But
no, he just talks a lot, which is fun.
Yeah.
And then in terms of Instagram
YouTube.
would be us. YouTube.
YouTube, definitely check out all the stuff you've done for Marco, which is fantastic.
Yes.
Bozeman Bio is going to be your go-to for all your lab resources. And he does some good statistics review because he does a lot of other subjects besides bio too. So he's a good resource for that.
And so we will come back together as you and me doing our Unit 8 live in 2 weeks. So that's that May 1st, May 2nd.
That sounds right.
May 2nd, we'll be back together.
Yep.
If you're wanting some help with math, I'm doing a full math with Marco Learning on May 1st. I'm going to take the formula sheet and literally work a problem from each equation on the formula sheet. My brain was not working today, as we see. I've already done, what, 2 lives today?
Two lives today.
So with all that, I would like to say good night, y'all. Remember your AP Bio Penguin, your dress of success.
Have a great night, guys. See you later.
Bye.